Hi everyone, my name is David Nicewicz. I'm a professor of chemistry here at UNC Chapel Hill. Like you, I was also a student here at UNC. I completed my PhD studies in 2006 and then started my independent career here as a professor of chemistry in 2009 and studying organic chemistry. So my laboratory is involved in this area we call photoredox catalysis. Our work is really focusing on designing new chemical reactions that are powered by visible light. This is pretty similar to how nature uses sunlight in a process, of course, called photosynthesis to harvest carbon dioxide in water using sunlight and turns this into carbohydrates and ultimately oxygen. So what we want to do is actually mimic that process to make fine chemicals that are important for modern society in a much more environmentally benign way. My laboratory has been successful in using organic photoredox catalysis to develop a number of new transformations, particularly the anti-Markovnikov addition of acids to alkenes. And this essentially inverts the normal regioselectivity of the addition of the acid to simple olefin substrates. But perhaps more excitingly, we've also been able to develop an oxidative functionalization of aromatic compounds with amines and ultimately to produce aniline-like structures. These are very common structures in a lot of different pharmaceutical agents, and I think perhaps most exciting is that the pharmaceutical industry is using some of these reactions to produce many different libraries of compounds that hopefully will eventually lead to the discovery of new drugs. The most important component of these reactions is the organic dye or photoredox catalyst that we use. They have a lot of different structural features as well as electronic features. And actually predicting which catalyst is the most ideal for a given transformation is quite difficult. So this is where you all come in. First, you'll be synthesizing these dyes. Secondly, you'll be characterizing them through a number of different techniques. And third, we hope to take these structurally different and electronically different catalysts and compare them to observe possible trends in terms of their structure and electronic function. And really, I think this will be a great introduction in terms of how modern organic synthesis and research is conducted. And I'm going to leave it right now with one of my graduate students who will tell you a little bit more about the project that you're going to do.
Hello everyone, my name is Cole Cruz and I'm a graduate student working in Dr. Nicewicz's lab. As he mentioned, I'm going to give you guys some insight into the big picture of some of our work and get you introduced to some of the concepts that you will be using during this research project. We're going to have you all synthesize and characterize some beryllium dye derivatives that we are interested in using in our lab. As we've mentioned before, the most important thing that we need to remember is that light is a source of energy. Photosynthesis is the process where carbon dioxide and water are converted into sugars and oxygen when plants absorb light. This is a chemical reaction, and like all reactions, it needs some energy in order to proceed. In the same way, the combustion of sugar and oxygen to form carbon dioxide and water is a chemical reaction that needs energy, and it's often supplied as heat from a fire. Remember that any reaction coordinate diagram features a reactant, a transition state, and a product. For any reaction to proceed, the reactants need energy to reach the higher energy transition state, to then go towards products. Our lab focuses on using light instead of heat to provide this energy. In order for a chemical to use light in a reaction, it has to be able to absorb it. Every molecule has its own absorption spectrum that dictates what wavelengths of light it absorbs. One molecule involved in the process of photosynthesis is chlorophyll A, and it has the absorption spectrum shown here. You'll notice that it absorbs much of the light in the visible region except for light around 500 nanometers, which helps give plants their green color. So if we wanted to make a plant undergo photosynthesis with our own light instead of sunlight, we would have to take into account the absorption spectrum of chlorophyll A. If we wanted to use only green light, we wouldn't be able to see a lot of photosynthesis because chlorophyll doesn't absorb that wavelength of light. If it doesn't absorb a wavelength of light, a chemical cannot use it in a chemical reaction. While chlorophyll A is one part of the photosynthetic process in plants, there are many other parts involved. One of the most important parts, actually, is this big structure known as photosystem II dimer. This protein has an extremely high molecular weight. While plants have become extremely efficient at synthesizing and using this protein for their own purposes, making it in the lab and using it as chemists would be pretty much impossible. Instead, we have turned to smaller organic molecules like these actinium and beryllium dyes. These simple and easily prepared dyes are what we use in our chemistry to try to use light energy the same way that photosystem 2 dimer does. In principle, we are mimicking the properties of these biological molecules with simple organic dyes. The most important property of these dyes is the difference in their orbital energy. This determines what wavelength of light they're able to absorb. The protein for photosynthesis, photosystem 2 dimer, absorbs visible light because the energy difference between its HOMO and its LUMO is matched with the energy of visible light. So like I said before, the wavelength of light that a molecule can absorb is based upon the energy difference between its HOMO and LUMO orbitals. So something that absorbs red light, for example, is actually absorbing light in the region of 620 to 750 nanometers. That wavelength corresponds to that energy difference in those orbitals. So, if the energy difference between those orbitals begins to increase, for example, we might expect that the wavelength of light that it absorbs would change. If it increases enough, you would start to absorb orange light instead, which is a higher energy light. Recall that the energy of light is actually inversely proportional to its wavelength. If we continue to increase the gap between the HOMO and the LUMO, the light that it absorbs will also begin to increase in energy until we go from yellow light to green light to blue light, and finally to violet light. Violet light is the highest energy visible light that we could use. Any light that's lower than 380 nanometers is ultraviolet light, and any light that is higher than 750 nanometers is infrared light. So we're working in the visible range within a 400 nanometer window of light. So what exactly is photoredox catalysis? We're trying to mimic photosynthesis by using light to allow new chemical reactions to proceed. This can happen because when our organic dyes absorb light, in many of our cases blue light, they enter an electronically excited state. In this state, the dyes become very powerful oxidizing agents, able to remove electrons from other organic molecules. The reason that excitation of a dye allows oxidation to take place can be understood using a simple molecular orbital diagram. Our dye has 2 electrons in its HOMO orbital and no electrons in its LUMO. Upon absorbing light of the correct wavelength, an electron is promoted from the HOMO to the LUMO, creating the excited state dye. This excitation has left an electron hole in the HOMO of the dye. Ideally, a stable organic molecule will have 2 electrons paired in its HOMO orbital. Since excitation disrupts this stable state, the excited molecule is very reactive because it needs to pair off the electron that's left in the HOMO. One way that it can accomplish this is by accepting an electron from another organic molecule. When a molecule like this alkene, with a HOMO at a suitable energy level, encounters the excited dye, it can donate an electron and become oxidized. This electron transfer process creates the reduced form of the beryllium dye and creates the oxidized form of the alkene, the cation radical. This cation radical is very reactive and can act as an electrophile. We have used this reactivity in many ways in our lab. In any photoredox catalyzed process, the most important step is the redox event, where the electron is transferred between the photoredox dye and the organic molecule. In order to determine when this electron transfer process is favorable, we need to know something about the relative energies of the HOMO electrons for these molecules. One way that we can quantify these energies is through reduction potentials. You should remember that electron transfer processes are generally categorized in terms of their reduction potentials. This value determines the ease with which it gains or loses an electron. When thinking about oxidation of an organic molecule with an excited state dye, we need to do a little math. We take into account the reduction potential, E1/2, of the excited state dye, which is the electron acceptor, A, and the oxidation potential of the organic molecule, the electron donor, D. Determining if the free energy of this electron transfer process is favorable is done by simply subtracting the reduction potential of the excited dye from the oxidation potential of the organic molecule. When the value from this calculation is negative, the electron transfer is exothermic and should proceed easily. In order for this value to be negative, the reduction potential of the excited dye must be greater than the potential of the organic molecule. The oxidation potentials of organic molecules are easily determined through a process called cyclic voltammetry. In order to do these calculations, we need to be able to measure excited state reduction potentials of our dyes, which is a bit more complicated. The excited state reduction potential of a dye is highly dependent on the structure of the dye. Simply by replacing some of the hydrogens on the benzene ring with methoxy groups, we can change the excited state potentials of these dyes by about 1 volt. As a result, our lab has become interested in varying the substitution on these berylliums to be able to create different dyes with specific properties. This is where you all will be helping us with this goal. You will be synthesizing new beryllium dyes and characterizing them so that we can determine their excited state reduction potentials. To do so, we need to know the dye's absorption and emission characteristics, as well as its ground state reduction potential. One way that you can think about the excited state reduction potential is as the summation of both the electrochemical and photochemical energies of a given dye. This is reflected in the equation that you will use to calculate this value. E1/2 refers to the ground state electrical energy of the dye, while E00 refers to the photochemical energy of the dye. E00 is essentially a measurement of the HOMO-LUMO energy gap. So you will be synthesizing derivatives of these dyes that have different substitution in the R groups. We are expecting that changing these R groups will change both the electrical and the photochemical properties of the dye. You would imagine that having either electron donating or withdrawing groups might alter these properties. We just don't know what effect they will have, and if they'll have the same effect on both parts of the equation. Ideally, if we make enough of these dyes and characterize them, we might be able to pinpoint which substituents have the biggest effect and can better design dyes for specific applications.
Okay, so hopefully that gives you an idea of what you'll be doing in the next few weeks, and also the importance of why you're conducting this research. And this gives you a glimpse, I think, into how we as scientists develop systematically new areas of research. Beyond applications even to the field of organic synthesis, the organic dyes that you make could also have applications to organic solar cell technology. And I think that's the important thing to remember about research, is that you never quite can predict maybe the path that you take, and ultimately some exciting results can come from unexpected sources. Remember that this is a collaborative effort, and ultimately you'll be working in small groups. And that's much the same way that my laboratory conducts its research. We work in groups towards a common effort. Definitely take careful notes. While you may not end up in an organic chemistry laboratory as your ultimate career, hopefully this will give you an idea of what scientists do to really push the boundaries of research. Just remember that if anything interesting comes from one of the organic photoredox dyes that you all synthesize, you'll be included as an author on a publication that comes from my laboratory that you can certainly cite on your resume. So I hope you enjoy the next several weeks, and thank you for all your efforts, and we look forward to seeing some exciting results.